In this video we will discuss about bacterial mediated cancer immunotherapy. So in this case we will discuss about the similarities of this T cell therapy and the bacterial mediated cancer immunotherapy, and one of the discoveries that enables to use bacteria for this cancer immunotherapies. So we will start with this adaptive T cell therapies. So in this case, before going into that, we will understand what the discoveries has been identified. This one of the discoveries that has identified is that bacteria can able to mimic the cancer antigen when they are co-cultured with the cancers, and similar phenomena has been observed for this bacteriophage. So the idea is that once this bacteria and bacteriophage can co-culture with the blood, so that the bacteria can have the antigens of the cancer cells. Similarly, bacteriophage also has the antigens for the cancer cells. Then the T cells can be isolated from this, and it can be isolated from the blood, and it can be engineered against the cross-reactive bacterial antigens. And thereafter, those T cells will be expanded in the ex-vivo conditions or in the in vitro conditions, and these T cells will then go through this drug development process, where the T cells can target the cancers against which the T cells has been designed using the cross-reactive bacterial antigens. And one of the streamline approach for this is that once we have this bacterial drug product libraries, and then we can streamline the process of manufacturing and increasing the mass production and safety and stability. Thereafter we will do the clinical test trials, and then make the bacterial drugs, and then use it for the drug development and for the patient treatment. With the development of the AI, we can modify this traditional approach so that we will have a much better efficient immunotherapeutic results. That is, we will have this AI based bacterial mediated cancer immunotherapies. So we will have those predictions based on the data that we get from the patients who are treated with this BCIT before, and their tumor characteristics and their sequencing data. And based on this data, this AI can predict which tumor antigens are known to have better cancer immunotherapeutic effect. And based on that, and also based on the patient who is going to be treated with this BCIT, it can design a personalized drug selection based on the antigens that are being determined in the patient, and based on the previous results that is presented in the form of data. So based on these two combinations and the AI predictions, it can predict the personalized drug selections which can have a optimized immunotherapeutic response against the cancers. And simultaneously, the AI based data can be updated with these bacterial drug product libraries, and these drug product libraries can be made by taking out the blood from the cancer affected patient, and then make the bacterial drug libraries. So this will give us the information what are the different kind of antigens that are present within the patient. And so this will enable us for the customized T cells based cancer immunotherapy. And once we have developed this kind of systems, then we can move on to this multidisciplinary approach, which will include oncologist, radiologist, nuclear medicine physicians, so that we can further improve the cancer immunotherapies to a point that once the patients are treated with this cancer immunotherapies, there is no redevelopment of the cancers in the patients at the later stage of their life. So this is the main goal. And right now I think some of the some of the drugs are in clinical stage for this panel A based therapeutic approach, but you can refer to the papers that has some of the drugs. There's a list of the drugs that are in the clinical stage, and they belongs to like which category. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.